So now we're above 50. So thank you all for coming to the Department of Biological Sciences annual meet and greet. This is our second year of doing this virtually, which last year we had to. This year we just decided it was a good idea so we could get as many people here as possible, no matter where they are physically at the moment. So I hope you guys will get a good chance to get a sense for the vibe of our department, meet a lot of faculty, get to know a lot of the grad students, hear something about some of the student organizations that are active in the department, and get any questions that you have answered about being a biology student as well. So I'm just gonna give it another minute or two for people to join, and then we'll go ahead and get started. And if you guys have any questions as we're going along, you can go ahead and type those in the chat too. So I'm gonna be the MC for today, but all of the faculty I introduce will be sharing their own screens to share their research and teaching. So we'll kind of be jumping in and out. But we've all become Zoom masters at this point. So. Okay, so I think we can go ahead and get started. So first of all, again, welcome to the annual biology meet and greet. Welcome to DePaul, welcome to the Department of Biology. These are just some scenes of some of our favorite spaces and our favorite things to do. General biology lab, hanging out in the atrium, checking out some exposure to the health sciences, but also to all sorts of other aspects of biology. And then obviously our flagship building. So I'm gonna sort of run through the people that make our department as great as it is. So we'll talk about research faculty, we'll talk about teaching faculty, about the graduate students that really do a lot of the amazing research that's happening on campus, and also TA for our courses, talk a little bit about some student organizations, and then also some support structures in place in the department that you may or may not be aware of yet. Okay, so I'm going to start with, this is just a montage of some of our many research faculty. Like I said, I'm going to let them do the driving from here on out, but we've got a really great breadth of work going on in the department, from cell and molecular biology to microbiology, evolution and ecology, immunology, neuroscience. So I think no matter what your niche is, no matter what your area of biology interest is, you'll be able to find some great work going on in the department and some great courses to get involved with as well. So I'll go ahead and stop my share. And I did get RSVPs from a handful of faculty. If I didn't get an RSVP from you, but you are here, if I skip you, feel free to jump in. But it's the first person that I know was planning to attend is, well, let's see, oh, I see Windsor here. All right, so we'll go alphabetically. So Windsor, if you have anything you'd like to share, you're welcome to share your screen.
Sure, thank you. Can you hear me?
Yes.
Okay, great. Hi everyone, it's very nice to see your electronic images. And so I'm Dr. Windsor Aguilar. I'm an evolutionary biologist and associate professor. I've been here since 2009. So there's an image on the screen. This is not me. This is John Avis, another evolutionary biologist and population geneticist. But he captured in an essay how I've felt about biology for a while now, in that it seems like these are in many ways the best and but also in some ways the worst times of biology. The best because, I'm sorry here. The best because if you ask, you'll see the faculty explain their research, and if you ask them about the things they're doing, they'll probably go on about all the exciting developments in terms of the methods that are available, the conceptual insights. And so we have now, today, the most extraordinary set of tools and the greatest accumulation of knowledge that we've ever had. This is my favorite. This is because I work in using molecular markers, this is a nanopore sequencer. And this little machine fits in the palm of your hand. It connects via USB to a normal computer, but it's a genome sequencer. So it could generate several human genomes worth of DNA data in a single run. They're portable, and the starter pack only costs $1,000, which is nothing from a scientific research perspective. So it's really an extraordinary little device, but it's like that in every field of biology. In the geographic information services, from morphological perspectives, data analysis, there's all these developments. So we've never had tools like this. But it's also, there's also an extraordinary need for biologists because in many ways, these are also the worst of times. The planet's never been in worse shape. Extinction rates are increasing. Humans are affecting every corner of the planet now, from the deepest parts of the oceans to the farthest reaches of rainforests in the middle of South America. So the effects of human population growth and overuse of the environment are at its worst. And the same can go being said for diseases like the pandemic that we have. So there's this kind of contrast of extraordinary set of tools and developments with also this really extraordinary need in terms of young people that can help us solve these problems scientifically. I'm an evolutionary biologist, and so my lab tries to understand how organisms adapt to changes in the environment. Natural changes, natural gradients, but increasingly we're studying changes caused by humans. And so we try to do research in many different, on many different topics, but the two primary areas that we study is the evolution of form. We're interested in how organisms change genetically as they adapt to different changes in environmental conditions, along elevational gradients or along gradients of human disturbance. And so we use geometric morphometric methods and other methods to quantify and model that change. We also use DNA sequence and molecular markers to get at the evolutionary history of species. So, we work primarily with freshwater fish. I teach a number of classes where I'm teaching evolution now, General Biology 2 in the winter, Concepts in Evolution, Molecular Methods in Ecology and Evolution is coming back in the spring. Biostatistics, Evolution, Health, and Medicine, so a broad array of classes. But I did want to take a second to make a special announcement about a new study abroad course that is scheduled to run in the spring break, Biodiversity and the Modern Extinction Crisis. We'll basically travel to Ecuador in South America and survey ecosystems from the high Andes to the rainforest region, one of the most biodiverse spots on Earth, and then to the Machalilla National Park on the coast of Ecuador. And in each of these areas, we'll learn about the biodiversity, how it originated, what are the the key species, and also the problems, the challenges. Oil extraction underneath one of the most biodiverse spots on Earth in the Yosemite Forest, deforestation and transformation of ecosystems, pollution, ocean acidification. So we'll talk about these as we survey the ecosystem. So this is going to run in the spring break. It's for first-year students. So it's a focal point seminar for part of the first-year program. The course will be in the winter, and then we'll travel over 10 days in the spring break. There's another study abroad program, Galapagos Evolution Society. This runs in the summers, so the course is in the spring, and then we go to Ecuador for a few days, mainland Ecuador, and then we go to the Galapagos Islands for a week, and we cruise around on a ship on different islands, learning about how the theory of evolution by natural selection was developed and why evolution matters for society. So this ran a couple of years ago, and unfortunately hasn't run since because of the pandemic. but we're excited to run it again with Dr. Dan Cohn, who is retired, but not retired enough to not do this course. And finally, last pitch, ¿hablas español? If you speak Spanish, if you can read and write Spanish, I'd be very interested in having you talk to me, so maybe we could talk about research possibilities. Much of my research is in Northwestern South America, but most of our papers have been in English until very recently. We're changing that. So I'm very eager to bring in Spanish speakers that can help me find the literature that's in Spanish and also translate a lot of our research so we can publish it in Spanish. So if you are interested in Spanish, and especially ecology, evolution, or computational biology, please do approach me. You are needed in my lab. Thank you.
All right, thank you, Windsor. And next up, we got Dr. Margaret Bell.
Unmute myself. Hi, everybody. Thanks for joining us today. Dr. Gear, I'm really glad you went first. That was an awesome introduction to our need to do science. Right now, I don't have quite as cool a spiel, but my interests are kind of an intersection of several complementary fields. My training's in neuroscience, but I'm also interested in understanding how hormones and immune processes can affect the developing brain. So we use a rodent model because it's hard to look at little kiddo models and understand their kid brains to understand at a molecular level what they're doing. But we are looking at animals that are exposed to an environmental contaminant and then understanding how that's shifting their developmental trajectory so that it could impact a range of mental health related, neurodegenerative related disorders. So we approach this from a couple different perspectives. We can use histology to understand brain structures and immunohistochemistry to understand what cells are expressing different proteins. We can study animals' behavior. We have a couple cool behavior testing setups in the facility. And we can understand what the cells are doing in an in vitro model. So we're trying to kind of span the gamut of ways in which we can answer questions about how environmental contaminants are affecting the brain. And I should also have backed up that my overall interests are how the environment as a whole is impacting the brain. And we're picking one environmental contaminant just as a way to start asking that question. The contaminants we talk about are called polychlorinated biphenyls. They're really common. Everybody has a body burden. They're especially common in Chicago, so there's some relevance there. But the concepts that we're learning with PCBs are probably also related to a host of other contaminants like diesel exhaust particulates, bisphenols, you've heard of BPA, related families. And really I'm interested in understanding all of the potential stressors or things that can push your homeostasis one way or the other in the environment. So it's part of a kind of a bigger picture. With that in mind, my teaching kind of makes sense, which is good. I teach a range of classes. I'm also a joint appointment in health sciences, so I teach some health science classes. For bio, I teach cell biology. I'm teaching it right now. I'll probably teach it once or twice every year. I also teach endocrinology, intro to endo. That's bio 386. That's on the books. I think it's scheduled for the spring. I also teach some 191 labs. I teach an intro or non-majors, brain and behavior. And then I teach a new class that's now going to be offered to biology students. It's called the physiology of poverty. And that's not a super accurate term because really I'm talking about socioeconomic inequality. And inextricable to that, at least in the United States, is racism. So we're thinking about psychological stressors of living in our society paired with other stressors like, or let's say challenges like access to healthcare, exposure to contaminants, sleep disruption, dietary challenges that all kind of interact to affect a developing brain and a healthy adult brain. So what physiological issues you see as a result of that. So that's going to be offered in the winter. So it's cross-listed Health 325 with Bio 225. With health sciences students there, we also hopefully have a December public health people. So I'm really hoping it's a cool conversation between people who approach these problems from different perspectives. So come at it from wherever you are. And then I teach Intro to Health Sciences, which is a new class. So I think that's what I wanted to say. My contact information, you can find us online. We have a lab website that you can find on the bio research page. It's linked through there. Would love to chat, get you involved. Thanks.
All right, next up is Dr. Joanna Brooke.
Can everyone hear me okay? Okay, all right, sorry about that delay there. So my name is Dr. Brooke. I am a microbiologist by training, and you can see the list of courses here that I teach. So microbiology right now, principles of biotechnology in the winter, and topics in medical bacteriology or topics in microbiology and biotechnology in the spring. So as you can see, my interest is in the area of microbiology. In terms, for some reason it's not going forward. In terms of my research, my area of interest is in infectious disease. I work with an infectious opportunistic pathogen. It's a bacterium called Stenotrophomonas multiphilia. This is an organism that causes respiratory tract infections, in addition to a variety of infections in humans. It is of significant concern to those that are susceptible and immunocompromised patients. And so one of the things that is of concern to physicians is the fact that this is a multi-drug resistant bacterium. And the rates of antibiotic resistance in this organism are on the increase. And so we're looking for alternative ways to combat this pathogen. The other point to make about Stenotrophomonas is it has the ability to form bacterial films on surfaces, and those surfaces can act as source of infections. And so in my lab, we are interested in understanding the genetic mechanisms underlying the organism's ability to cause disease, and also to look for alternative strategies to prevent infection and fight infection by this organism. I mentioned lab members to remind myself to say, yes, I work with a variety of graduate students and undergraduate students. So if you're interested in the research I do, my contact information is there, so please contact me. I'm always looking for helpful brains in the lab, so your input is welcome.
All right, thank you very much.
Welcome.
Jason Vistrianski.
All right, hi everyone. Can everyone see my screen okay? I'm gonna throw it into slideshow mode here. Okay, so I'm Jason Vistrianski. I'm an associate professor here at DePaul in biology. My background is I have an undergrad in marine biology and a PhD in what's called comparative physiology. Essentially, I study how organisms work and how they deal with challenges in their environment. So some of you may have already had me in a course or two. I typically teach the 3rd Gen Bio course, so Bio 193. I usually teach that with Dr. Dean. I teach the animal diversity section. I also teach a marine biology course for non-majors. And then most likely you'll find me in one of the upper level physiology courses. So either Bio 307, animal physiology, or 352, advanced comparative physiology. I also teach a research-based study abroad program in Spain, ideally every summer. We haven't been able to do it the last couple of years for obvious reasons. But the plan is to go ahead with this program next summer. So if you're interested in getting research experience and learning how to do research, Dr. Grice in chemistry and I developed this 2-course study abroad program in order to get students experience working in research. So, for a lot of our students, it's their first taste of research. And after that, they hopefully go and find their way into different faculty labs and get some more experience that way. So a little bit more about what I do. As I mentioned, I'm a comparative physiologist. This is a picture of me from grad school holding an Arctic char, which is a salmonid species of fish from the Arctic coast. And what I'm most interested in is how different animals are able to live and survive in changing salinity. So you may know that a lot of animal species are able to migrate from either freshwater to seawater or vice versa. And for those animals, it's a very challenging situation. Think about the last time you were out down at the ocean and got a gulp of seawater, how uncomfortable that was. Well, just imagine all of the animals in the sea drink seawater as their main source of water. So somehow they're able to deal with such a high dietary salt load and not die like we would. So I study the physiological basis for why those animals can survive in high salinity oceanic environments, but also the opposite. How do animals deal with very low salinity environments like Lake Michigan or streams and rivers? And so I'm studying mainly fishes because those are a great group to study this, because most of the animals that can migrate back and forth are in that group. And I specifically study their gills because that's the organ that they predominantly use to balance the salts in their body. So humans predominantly use our kidneys to balance salts, and fishes use their gills. So it's a very interesting comparative type question I'm asking. How do animals deal with changing salinity? So having said that, that's what I do. I'm always looking for volunteers to start in the lab and maybe hopefully work for more than a few quarters, maybe a year or two in the lab. So if you're, especially if in your first or second year and you're interested in getting into research, let me know. We can talk about opportunities in my lab or the study abroad course that I'll teach coming this summer. Thank you.
Okay. I'm going to briefly jump ahead in the alphabet and introduce Dr. Eric Norstrom.
Thank you. Let me share my screen here. Sorry, as you can see, I'm downtown. It's a little hard to, just kidding. I don't usually use backgrounds, and I'm sharing. And can you see my little laser pointer? You guys can see that? And no? Yes, you can. Okay. Well, thanks for coming by. It's great to see everybody's virtual icons there. And this will be quick. It's all on one slide here. That's me up in the corner. I'm a neurobiologist. So my PhD is in neurobiology, and my focus is neurodegenerative disease. So I study specifically Alzheimer's disease, and a little bit of others, but primarily Alzheimer's disease. So that's characterized by the degeneration, a progressive degeneration of the brain, really starting in these areas associated with memory formation. And you're probably familiar with the kind of classic presentation, clinical presentation of Alzheimer's with dementia and loss of memory and personality changes. So my focus is in the world of the extremely small, really submicroscopic structures. And I'm really interested in proteins. And the reason for that is that the Alzheimer's disease is a primary pathological and pathogenic component is the accumulation of these small peptides that are hydrophobic. In the brain of an Alzheimer's patient, you see two obligatory pathologies for this diagnosis. One are these tangles, which are made of a different protein that I don't study. But what you see here are what we call plaques. And these plaques are composed primarily of just a small little peptide that's only 42 amino acids long, 40 or 42. And that little peptide is liberated from a large protein, a very 70 kilodalton protein by enzymes that chop that protein up and release this peptide. So actually what I study are the proteins that interact with this large, what we call precursor protein, and that can bias its metabolism toward generation or not generation of this A-beta peptide. Generation of this peptide is not the only metabolic fate for this protein. So I'm interested in what influences that metabolic fate. And to do that, primarily what I'm doing on a day-to-day basis is protein biochemistry. So I look at where proteins are in cells, what conformations they're in, how they're associated with membranes, and how they interact with and influence the metabolism of this precursor protein that yields this pathogenic peptide. Yeah, so that's what I do. It's kind of lab ratty stuff, not exciting, beautiful vistas like like some folks get to see, but all the same, it's very cool to see the results we get. And here's what I teach. You might see me, I mean, mostly what I'm teaching is human anatomy and cellular neurobiology, and because that takes up a lot of my teaching load. And occasionally you might see me in Gen Bio or Introduction to Neuroscience. So I'm also the director of the neuroscience program. Of course, as bio majors, you have a great way to study neuroscience through the neuroscience concentration, which I also am in charge of, but I'm also the director of the neuroscience program. So I teach a little bit for that and do research whenever I can. It's my favorite thing to do, but unfortunately, with the administrative appointments, it's very difficult, and you have to be really organized to do it. So yeah, that's me in a nutshell. Sorry, I didn't put my contact info there, but I'm gonna stop sharing and I'll put it in the chat. And feel free to contact me about anything. Really, I'm always happy to talk about anything. So there it is, and that's me. Thank you so much for listening.
Right.
Oh, somebody asked, sorry, there was a question about, is Alzheimer's disease inheritable? Yes, it can be. It can run in families. There are specific mutations that can lead to inheritance of Alzheimer's with varying levels of penetrance. That is to say, some mutations only influence your probability of getting Alzheimer's, whereas other mutations are extremely aggressive and can cause Alzheimer's at very early ages, believe it or not. And interestingly, every mutation associated with acquiring Alzheimer's disease increases the levels of those peptides that I talked about. So that's the real link between that. Thank you for that question. Thanks.
Okay. Next up, Dr. Sarah Connolly.
Here we go. All right. So I'm Sarah Connolly. I'm an associate professor in biology, but I have a joint appointment in health sciences as well. I'm up here in McGowan South, and my lab and office are both up in McGowan South on the 4th floor. I teach, sorry, there's a window covering my screen. There we go. I teach Bio 191, Gen Bio 1, as well as Bio 210 microbiology. I have taught molecular virology. It's a course that's offered through health, but it does count as credit for a bio elective. I'm not teaching it this year, but perhaps in future years. And then I've taught a few courses in health sciences, as well as a freshman seminar. I study viruses. So specifically, I study how viruses enter cells. I use herpes viruses as my model virus. There are 9 human herpes viruses, and most people are infected with more than one. Some people know that, some people don't. What I study is how we get from this first picture here, which is a virus particle bound to a cell, to the second picture. How does the fusion occur? How does the fusion occur between the virus and the cell to allow the virus to drop off its genetic information inside the cell? And the entry mechanism is conserved among all herpes viruses. So what we learn about entry for one herpes virus is going to be relevant to all the herpes viruses. And what we learn about fusion in general is going to be relevant to all of the viruses that have a lipid envelope layer. So this includes influenza and Ebola, rabies, HIV, smallpox, and SARS-CoV-2. I have 2 slides. This is my last slide, but I can sort of give you a pitch as to why viruses are fun to study. So they're very simple. They are just a small genome. Some viruses have only 12 genes, surrounded by a protein coat to protect that genome. It can be an RNA or a DNA genome, but a genome and a protein coat, that's all some viruses have. And then some other viruses have a lipid envelope layer. But that's it. There's no metabolism, there's no mitochondria, nothing. But these viruses are so powerful, they can take over host cells. They can even shut off host cell expression and make the cell just make the viral proteins. And as we've seen, viruses can spread to, through an individual as well as through a population. So there are generally 4 overall steps to a virus life cycle. The first step is binding to a cell and entering it. And then the genome is replicated in the cell, the viral proteins are made, and the viruses are assembled, and then they exit the cell. And the part that I study is this first step, the attachment and entry. So this is mediated by proteins that are in that lipid envelope. So we've all heard about SARS-CoV-2 spike. These would be the proteins in herpes viruses that are analogous to that spike protein. I study how those proteins interact with one another to trigger fusion, and I study how the actual fusion protein undergoes this massive refolding event in order to cause the viral envelope to fuse with the cell membrane. That fusion event requires energy, and it's the interaction of these proteins with each other as well as with the cell that provides the energy for that fusion event. One of the reasons to study virus entry is that it's a really nice target for antiviral intervention. So if you can stop the virus from ever getting into the cell, you can stop the infection overall. So the techniques I use in my lab are cell biology, molecular biology, protein biochemistry, and some virology techniques. And if you're interested in studying herpes viruses, send me an email and we can talk.
All right, thank you very much. Now we have a special guest who's not only a member of the biology faculty, but also the Dean of the College of Science and Health. So welcome, Dr. Stephanie Dance-Barnes.
Hey everyone, I hope everyone's having a great day, and it's definitely been a busy Friday for me, and I'm sure it's been busy for you. I first just want to talk about my vision for the upcoming year, and it is to continue to focus on some of the strategic priorities of the college, and there are some things that are already underway in regards to ensuring that we're providing quality programming for our undergraduates and graduates. And that part of that does encompass ensuring that we're fostering a culture where we're promoting opportunities for undergraduate and graduate research. There have also been much work and continuing work being done to expand and promote our DEI efforts. And more to come with that. We've already done some reorganization in the Dean's office, where we'll have dedicated funds for initiatives, as well as various staff that will be aligned to help support those particular efforts. Additionally, I want to make sure that we're communicating some of our research and innovations more robustly. And so we're hopefully going to be doing some advancement with some of the website and web page promotion. So more to come with that as well. And then there will be more strategic efforts being put towards how we're managing retention and also persistence and student success. And so I'm very excited about many of the opportunities that I think are coming up in the very near future. And I guess first and foremost, I just want to say, please understand I have an open door or open Zoom policy. So if there are any questions, comments, concerns, or feedback, please try to get on my calendar, and I would definitely try to connect with you and have those meaningful discussions. I have prepared, and I think probably need to go the screen way. So I have prepared a slide, and hopefully it's not overly complicated, but my work does center around breast cancer, primarily triple negative breast cancer. And what I have done for the past several years is attempt to create a lab that does accommodate undergraduate students, particularly those that may not have had a lot of opportunities or experiences with research. And so I've tried very hard to create an environment that fosters opportunities for innovation and inquiry within my lab. Triple negative breast cancer, it is a breast cancer that's very prevalent, particularly in young African-American women specifically, but it is a very aggressive cancer that is found to be very difficult to treat because it does not have those typical receptors that most traditional therapies target, those being the estrogen, progesterone, and the human epidermal growth factor, or HER2. And so it's usually very difficult to treat these forms of cancer. What I'm going to show you very quickly are just some of the results or data or the types of projects that are typically done in my lab. And like I said, these have been catered or adjusted to help support undergraduate research experiences. And so, as I mentioned, my work primarily centers around cancer, and particularly breast cancer. And a lot of the experimentation looks at understanding the genetic changes involved in the initiation, promotion, progression of the cancer, and really exploiting some of those genetic changes with the hope of identifying targeted therapies. And so within my lab, some of the work that's been done by undergraduate students is around basic cell culture. So growth curves associated with particularly a novel breast cancer line that I developed that focuses on a triple negative breast cancer. It recapitulates triple negative breast cancer. And particularly with this cell line, it is BRCA1 and P53 deficient. And as you can see, it's a very aggressive cell line as compared to normal, and a more commonly studied breast cancer cell line, MCF7, in which the BRCA and P53 are wild type. Additionally, students have done BRDU or proliferation assays where they've examined, once again, this particular novel breast cancer cell line as compared to, for instance, if you're looking at the EPH4, this is a play on the normal cell line. And as you can see, is the K14, which is the novel cell line, is more proliferative in its activity. Another type of assay that students have developed in my lab have been a soft agar assay. And this kind of mimics what we would, as much as we can in culture, what the environment would be like for cancer cells, but not really. It just kind of mimics that. And what this does, it examines the anchorage independence of cells. And so, of course, cancer cells, they don't have to, they are independent of anchorage, where normal cells, they need to be adhered or anchored. And so what this assay allows for is for you to assess the tumorigicity of the various cancer cells. And as you can see here, the K14, which is the novel cell line, very much more tumorigenic than some of the other cell lines. And so I'm just showing you this to give you an indication of some of the types of assays and work that has been previously done within my lab, and the types of opportunities that are available for undergraduate and or graduate students. Additionally, we've looked at the ploidy status of the various cancer cells. So here, this is just showing you a karyotyping assay that students devised themselves. And what they were able to find was that this particular cell line, our novel cell line, was very aneuploidy in nature, which it had at abnormal levels or numbers of chromosomes. And then on the back end of that, we then looked at the ability to target these particular, some of the genetic changes that we see in this particular cell line, as I mentioned before, with P53 and BRCA1. And diallyl disulfide is an agent that's a natural agent that's been found to target P53 deficient cancer cells. And we actually tested that in this particular cell line. And as you can see, there was a 70% decrease in viability. And so I'll kind of stop there, but I wanted to also highlight some of the other types of experimentation that's been done by myself, and that I like to involve students with as well. And this was more of a study associated with involving 3rd through 5th grade students. And this is a current grant. And with this particular grant, we are creating a STEM learning ecosystem, which involves a buy-in from the school system and families and parents and STEM career partners. And essentially, we introduce 3rd through 5th graders to culturally relevant interventions to increase their, hopefully their interest in STEM and getting more information about their how they feel about their own science identity. And so I'll kind of stop there because I'm sure I've gone over my 5 minutes.
Well, thank you very much for making time to join us today. I know you have a very packed schedule, so I appreciate it.
No problem. Thank you guys for having me.
Okay. Next up on the alphabet is Dr. Bill Gilliland.
So hello, everyone. Let me bring up my slides here. Okay, can you see that? Okay, so I study the process of egg development in this organism here, the fruit fly Drosophila melanogaster. The ruler at the top of the screen here is in millimeters, so these are little 2 to 3 millimeter critters. And they are essentially chromosomes with wings. We use them as a convenient way to study chromosome movement. The abdomen of this critter is about 50% by volume ovaries, and those ovaries are full of eggs. And so to zoom in by about a factor of 10, here is 100 microns, which is 1/10 of a millimeter. And in this little rectangle superimposed here are the chromosomes. And so how these chromosomes move through the process of meiosis is the question that I'm trying to address. What genes are required to make the process go forward, and how do those work? And students in my lab will do both genetic experiments where you cross the flies and you examine the progeny, as well as cytological experiments where you prepare cells for viewing on the confocal microscope. The confocal microscope is an instrument that DePaul has. We are very lucky to have access to a good microscope, and I'm the director of it. And so I have another window here, which is an animation of some microscopy data that I have here. So it's going to show 2 molecules. One is actin, the other is DNA. So I'll just play the video here so you can see, and it rotates virtually, so this is computer interactions, and these little rings are cytoplasmic bridges between the nurse cells, and then the DNA here, the large blue circles, are the nuclei of the nurse cells. And when it finishes this animation, it'll do it again with both together, so you can see that these molecules are in different parts of the cell, and that the ring canals, which are those red circles, are connecting those nucleic cells. In addition to research, I also teach, and I teach Gen Bio this quarter, so I recognize a couple students here. Next quarter, I'll be teaching Bio 260. I've also taught part of the first year program, a seminar on cloning biotechnology, but I really want to talk about a new class that I'm going to be doing this spring, which the tentative title is Advanced Eukaryotic Genetics, and it's kind of a follow-on to Bio 260. Bio 260 is the classical genetics curriculum, and this class will explore the modern genetics curriculum. So things like whole genome sequencing, transcriptomics, proteomics, all of these high throughput tools that have really made this last decade the golden era of genetics. And what I'm really excited about this class is that it is going to be a course-based undergraduate research experience. I have joined a consortium called the Genomics Education Partnership, and the GEP does distributed science across around 100 institutions across the country. Using the power of massively parallel undergraduates, data is generated for whole genome sequencing in a central lab, and then the very labor-intensive task of annotating the genome is distributed to various institutions to be done by students in these courses. And so the lab for this new class will be gene annotation. You will get a chunk of de novo genome that has never been annotated before by anyone. And students will find where the genes are, where the transcript start and stop sites are, where the intron exon boundaries are, and what homologs, which versions of those genes are in this species compared to the reference genome of Drosophila melanogaster. If you complete this class, students also have the option for an additional small amount of work of being co-authors on the published papers that incorporate their data. And the consortium has already published a number of papers that have upwards of 400 co-authors, where these undergraduates got to contribute their work to real science that gets published in real journals, okay?
All right, thank you very much. I love the idea of massively parallel undergraduates. For some reason, that term just sounds fantastic. Okay, next up we have Dr. Jingjing Kip.
Let me mute myself. Hi everyone, I'm Jingjing Kip. Let me share my slides with you. Okay, so I'm professor from the biology department, and I'm in the field of physiology. Received my PhD from Department of Integrative and Molecular Physiology from Urbana-Champaign, U5. Since then, I did my postdoc training at Northwestern University, and then I joined DePaul. At DePaul, I teach a variety of physiology-related courses. And meanwhile, I also run a research lab focusing on molecular endocrinology. So let me give you an overview of the courses I've been teaching at DePaul, which you can see from this slide. This basically lists all the courses I taught throughout these years, but the top 5 are more recent courses that I've been teaching. I teach mostly physiology-related courses. I also developed a new course, which is called Mammalian Reproduction, since I started at DePaul, which is a seminar course and has been very popular. And currently, I also teach human physiology, which has a lab. It's for biomajor students. I've been teaching capstone seminars, and then starting this winter, we're going to have a new course, which is called Mammalian Physiology with lab sections. So you can see, basically, I teach physiology-related materials and reproduction-related materials. And in terms of research, as I just mentioned, I'm a molecular endocrinologist with a focus on reproductive science. And in particular, we are interested in the hormonal signaling and the gene regulation of ovarian follicle development. Some of our current focus include functions for vitamin A and calcium signaling. So vitamin A, as we know, is a nutrient, and in order for vitamin A to be effective in our body to facilitate development, vitamin A has to be degraded, break down into a chemical which is called retinoic acid. Retinoic acid then can enter the cell and bind to DNA and modify gene transcriptions. So we're interested in the retinoic acid signaling pathway and how this pathway may interact with some other signaling pathways and in the process of ovarian development. So this one, just a slide summarize some scope of the research we've been doing. As you can see, we do research at different levels, cellular levels, like intracellular levels, sorry. You can say like subcellular levels. We did live cell imaging, labeling to localize calcium image in the mitochondria, in the endoplasmic reticulum inside the cell, and to see how retinoic acid may regulate this kind of signaling pathway. We also perform cell proliferation, apoptosis, differentiation assays using cell cultures, either primary cells collected directly from animal tissues or cell lines that we have in the liquid nitrogen-free tank. And also we do tissue cultures. My students have done some follicle isolation from mouse ovaries, and also my lab perform a lot of animal research, in vivo studies, and currently we actually working on 2 new transgenic models that my lab have developed recent couple of years. And these 2 transgenic models basically knock out the retinoic acid receptors in the ovary only. And these 2 models are the only kinds of this model in the world, basically. So we're kind of very excited about this model, and we're in the process of further characterizing this model. This slide just summarizes some basic techniques we use: animal cell culture, morphology, histological study, and again, we're a molecular biology lab. We do a lot of DNA, RNA, protein studies, and we study live single cell imaging as well. And as I know that just like myself, I keep good relationship with my training institutions and the mentors. My lab also, continue our support to my students even after they graduate. Just a couple of days ago, I received an email from a previous undergraduate student who did research with me for 2.5 years. She is defending her PhD from Johns Hopkins next Wednesday, and then she got a postdoc offer from Harvard already. So she invited our whole lab to attend her thesis defense. So of course we will be there to continue to support her. So anyway, students from my lab, they move on to research or they move on to medical school. So these are the major directions. Of course, there are also people doing different other things. If you're interested in research opportunity from in my lab, here's my email, which would be the best way to contact me. And hope you enjoy the rest of the talk.
All right, thank you. So Dr. Keselowski, I didn't have you on my playlist, but I noticed you were here. Did you want to speak?
Sure. Hi, everyone. Sorry, I'm not live on video, nor do I have a PowerPoint because I am in my car. I am in Ohio driving to Parents Weekend for my son who's at Ohio State. So I apologize. I'm not super prepared, but I just wanted to be here to say hello. I'm Dorothy Keselowski. I have a joint appointment between biology and neuroscience. In biology, I teach behavioral neuroscience, I teach biostatistics, and I teach introduction to pharmacology. And then in neuroscience, I teach the capstone as well as a course on conversations with neuroscientists. And currently I'm teaching a course on neuroplasticity, which is a research topic that I'm interested in, which focuses on how the brain changes itself. So research-wise, I study traumatic brain injury and how the brain recovers. I'm a behavioral neuroscientist, so we use an animal model. We study the animal's behavior, and then we also study changes in the brain using histology. Current projects are looking at sex differences in subconcussion, and that's in collaboration with Dr. Bell. And then also looking at how the ApoE4 gene contributes to recovery post-traumatic brain injury in collaboration with Dr. Kudabak. And then I also do some research on the intersection of traumatic brain injury and domestic violence. So if you're interested in chatting with me, please reach out. And sorry for no visuals. Thank you.
You've got a good reason, I think. All right, next up is Dr. Jelaine LaMontagne. Sorry, I'm getting stumbling over my pronunciations this late in the Friday.
All right, I didn't unmute myself. Okay, hi everyone, I'm Dr. Jelaine LaMontagne. I'm an associate professor and I've been in the biology department since 2011. I'm a population ecologist, so my background, I have a bunch of degrees in ecology, and that also is reflected in my teaching. So right now I'm teaching ecology, which is about 215 in winter. I'm teaching part of 192, and then I also teach a research methods and applied biostatistics course. And in that course, students are learning how to analyze real data sets using computer programs. And it's a great sort of skills-based course. as well. There are a number of undergraduates who've gone on and gotten jobs and have led to their careers in data science and things based off of taking that course too. That sort of led them on that path. And I also teach population ecology. For my research, so I'm a population ecologist, so that means I'm interested in sort of how and why populations change over time and space. And one of the main systems that we study is reproduction in conifer trees, which is referred to as massed seeding. So every once in a while, these trees produce massive amounts of cones, and acorns on oak trees do this as well. But most years they have pretty low reproduction. And so if it's a high reproduction year, and it actually is in Chicago. So if you go around and look at conifer trees, they have lots of cones this year. And in the fall, they look all brown at the top because all these cones have sort of dried out and have opened up to release their seed. And I and some collaborators discovered that you can actually detect this using satellite imagery. So that's pretty cool because it happens usually over pretty large spatial scale. So my lab, we do a lot of field work for this research. We count cones on trees, we collect environmental data, and every once in a while, this is from this summer, I was out with some students and you take breaks and pick raspberries because that's a perk of field work. So a lot of my lab work is done in the field, but I also use really large data sets across the continental or global scale for a lot of the research that we do too. And this was some work that came out last year saying that these high reproductive events, when these trees look like this, sort of, if they are happening in Eastern North America, they tend to not be happening in Western North America. And those have lots of impacts for a variety of different species that feed on these seeds as well. We're also interested in studying climate change and how that's going to impact conifer tree reproduction. And this is a collaboration with the Oak Ridge National Laboratory that has an experiment called the Spruce Experiment that stands for Spruce and Peatland Response Under Changing Environments. And they have these huge environmental chambers that are set up out in the forest. You can see there's trees actually in there. And they have different temperature and carbon dioxide treatment. So this one says, welcome to a warmer future, plus 9 degrees Celsius and elevated CO2. And there's a grad student here, Teddy, who's working on this project and how the cone characteristics are changing and affecting seed numbers as well. Of course, all this work we do can affect the future of forests in terms of what trees are going to grow up afterwards. So here's a small seedling. Could affect mortality patterns across different species. Can impact a variety of different species of consumers. So here's a bird that actually has a spruce seed, white spruce seed in its beak there. And this all can have implications for disease, for human health of disease, because ticks have been shown to have relationships with these mass eating patterns. And so we're investigating that at continental scales as well. If you like hiking and doing research, this is one of the areas where we do research. This is up in the Upper Peninsula of Michigan. This is Lake Superior in the background. And this is one of the hikes that we can do when we're on our time off. But if you're like, I don't want to go out in the woods and you're interested in urban ecology, my lab does that as well. And one of the systems that we study is tree cavity availability. So here's grad student David Hull, who's looking at how tree cavities, either created by woodpeckers, this one's created by woodpeckers, or they can also occur from just branches falling off trees, how those have changed over time in different land use types within the city of Chicago, and also how these impact bird populations too. And red-headed woodpeckers is a species of concern in Illinois that also uses these cavities, and some of the work we've done has been focused on that too. But because I'm a population ecologist, I tend to study plants and animals. I'm interested in kind of all of it. And I have a grad student, this is Pizza Rat, who was famous a few years ago, looking at rat population dynamics across the city of Chicago. So that's my research. And if you're sort of a sophomore or junior and have an interest in ecology, please talk to me. We have some positions available in the lab. We bring some samples back from the field that need some processing. And I'm also the faculty advisor for the DePaul Ecology, Evolution, and Physiology Club, and they're a pretty active club. So if you have interest in ecology or evolution and physiology, I'd also encourage you to get in touch with them. Thank you.
All right. Dr. Kenshu Shimada.
Right. Can you guys hear me now? All right. So my name, well, let me see. Hold on a sec. Here we go. So my name is Kensho Shimada, and I'm a professor of biology, but I'm also a faculty of the environmental science and studies. So I got to teach only half of the time for biology, but when I teach, once in a while I teach general biology too. I teach, next quarter, I'll be teaching the topics in paleobiology class. That's 300, 400 level courses. So if you're junior or senior who needs to take those courses, and sometimes sophomores can take, please consider taking the course. I also teach comparative vertebrate anatomy class for biology every other year. I'm not scheduled to teach this year, but hopefully the following year. For environmental science, I teach for biology for environmental science, studies, as well as animal diversity class, which some biology students have taken the course for using electives and so forth. So research-wise, I'm a vertebrate paleontologist, although I sometimes use different titles depending upon where I'm at. I'm a comparative anatomist, I'm an evolutionary biologist, I'm a vertebrate zoologist, I'm a paleoecologist. I use different kinds of titles, but if I have to pick one, I'm a vertebrate paleontologist who studies ancient backbone animals. And particularly, I'm interested in the evolution of the marine ecosystem. And if you see the middle of the screen, you see the North America, but it doesn't look quite like a present-day North America. That's because this is the map of the North America during the age of dinosaurs. And during that time, there was a large ocean that crossed the continent. And you had to realize that the North America is divided into two parts. And both those land masses had dinosaurs. This is the dinosaur aged time. But what I'm really interested in is what was going on in the ocean. So I primarily study sharks, but in order to really understand the ecosystem, I also work on bony fishes, seabirds, marine reptiles, and anything that comes with the sharks. That's what I do. And also, as a comparative anatomist, I work on the modern day sharks as well. So on the left side of the screen, you will see some students dissecting the modern day sharks, and those information can be used in an evolutionary context or applied to the fossil record. But also I have many of the paleontology related topics, study of fossils. What I usually have, or many of the students in my lab have worked on examining the rock or sediment samples like collected from, say, Kansas. And I usually give a bag full of dirt essentially to a student, and a student will wash the sediments or break down the rocks and try to figure out what kind of fossils we can find, and then report all the different taxonomic diversity that we find for each rock unit. And those pieces of information can be eventually accumulated to talk about the change in the ecosystem over time due to environmental changes. For example, the ocean that I just told you, the one that runs in the middle of a continent, that ocean, there were times that were deeper, became deep and wide. Sometimes it became narrow and shallow. So we can talk about the climate change, the oceanic change, the environmental changes, and then how that the ecosystem will affect the organisms. So you see a lot of pictures of my former and present students, and I have Jake Wood and John Duranick as my graduate students currently. Right before the COVID hit, I had probably the most active time in my lab. I had 3 graduate students and 7 undergraduate students, but sadly, practically all of them graduated. And I'm really now looking for more, at this point, not more undergraduate students. So if you are, especially sophomore, junior, maybe senior, if you're interested in working with me, please shoot me an email and we can arrange a meeting and we can go from there. So thank you.
Okay. So they say to save the best for last. So coincidentally, alphabetically also, Dr. Tim Sparks is last of the research faculty, and he's also our brand new department chair. So please welcome Tim to his first year as chair, and we'll give him a chance to speak about that and also his teaching and research.
All right, well, thank you so much and welcome. And those of you that are freshmen, welcome to the poll. Those of you that are returning, welcome. And those of you that are returning for the first time, welcome to you also. So as Karen mentioned, I'm the new chair of the bio department. As I start talking, you're probably going to hear my accent's a bit strange. So I'm from the UK originally, South Wales in the UK, went to college in London, then went to grad school at the University of Kentucky. So I picked up a few Kentucky phrases here and then I might drop a y'all in the middle of a conversation. I came to Chicago in 2000. So I've been here for over 20 years now. I've actually lived in the States longer than I grew up in Britain. I moved to the States when I was 22. I started off doing population ecology research and then transitioned into more behavioral ecology research as a grad student. And now I work on behavioral ecology and parasitology. So if you ever seen any of those news stories about zombifying parasites that take over the control of the minds of the hosts and make them do crazy things, I work on that stuff. I work on some of those things locally and in other places, so these zombifying parasites. And these transitions through my career, I'm a first generation student in college. None of my family members, close or distant, went to college. That was a very new experience, and that had its own stresses and learning curves to go through, which many of you also probably may be going through or have gone through yourself. So I have some kind of insights in those places. And then as I went to grad school in Kentucky, I was an international student, a foreign student, right? So I have some of those kind of insights too. So I bring those into my teaching and research. Teaching-wise, I teach throughout the curriculum, but I teach less now as the chair. So you may see me in the Gen Bio sequence. I'll be teaching 192 in the spring if you're a freshman or a sophomore. If you're a higher level student, I might see you in the capstone class this year, or I'm teaching the intro to grad studies class for all the first year grad students. That's my teaching for this year. One thing I want to emphasize is you've just seen the number of diverse research faculty and diverse research opportunities that exist in the program. And before you come to the poll, that may not be as obvious just how much depth of research is going on, but there's an enormous depth of research going on in the department. And just over the we've always kind of known anecdotally, but over the last year or so, we've begun to really understand how important those research experiences are to students. So as should be clear from all these presentations and the options that may be available going forward, getting involved in research is a hugely beneficial experience for any student. And hopefully you'll be able to pursue that, whether it's through a CURE-based course, as Dr. Gilliland mentioned, or Dr. Vistrianski mentioned, or through individual labs. And going forward, we're going to try to create more of these, as Dr. Dan Spahns mentioned. Those are great experiences to get. So trying to get yourself involved, connected to research experiences is an amazing opportunity, amazing thing to do. On top of that, we've got this diversity of courses that we just mentioned, and then the diversity of programs that are developed and the diversity of faculty. All those things together giving you all the opportunities you could possibly imagine if you take advantage of them. And one of the things we often see the students, they maybe don't take full advantage of all the things available. Take advantage. We will do everything we can to assist you, connect you, and support you in all of these different options that you may want to pursue, but we're not going to force you, right? So we're going to really just do everything we can to provide a friendly, warm, and welcoming context and culture for you, but take advantage of those resources. When I teach the capstone class, which is the seniors, I often ask the students, if you could go back and be a freshman again, what would you do differently? And they almost always say, I wish I'd taken advantage of all of those resources. I waited too long, or I waited till I was a junior to do that. So take advantage of the resources. We all want to help. We all want to support you. If you ever feel overwhelmed, you feel anxious, which can happen, just reach out to us and we will always help you. Just like Dr. Dan Spahns, I have an open door policy. My office is in McGowan North in the main office. Stop by and see me. Stop by and talk to me, email me, whatever. I'll be happy to talk to you about any of the different things you may be interested in, programs you're interested in, research opportunities we may be able to try to get for you, or internships. All those are things we will try to do to help you find what you want to do so that you can be successful the way that you define success. So how you want to be successful, what is you want to do, what your passion is, we're going to try to help you navigate that. So we do that by doing a combination of knowledge that you expect to get in a science degree, transferable skills which you pick up in that science degree, and possibly most importantly, empowering you to be self-confident to navigate your own path with our support. And that's our goal, is to get you to the place where you're confident, empowered, knowledgeable, and have a skill set that can allow you to do anything you want to do. With our open door policies and our friendly supportive structure, you can achieve all of those goals. So with that in mind, thank you, welcome, and I hope to get to meet all of you as you go through your careers.
Okay, thank you very much to all the research faculty for making the time to share their teaching and research today. I think Dr. Bell early on had dropped something in the chat with a link to everyone's information in case you wanted to track anyone down. I just repasted that in the chat because there were also some faculty that were not able to attend today. So I would encourage you guys to all check out that page to hear more, to find out more about the faculty that did speak today and also learn about some of the faculty that weren't able to join us. I'm just going to share my screen again and talk about some of the other faculty in the department that don't necessarily have a research lab that you can join, but certainly involve students in their work as well. So lab coordinators like myself often will hire undergraduates to help with some of the prep that goes into getting materials ready for our bigger teaching labs. And then we also have quite a large contribution of teaching faculty that not only work with non-majors courses with the general biology courses, but also teach specialty courses of their own. And we'll all give a little bit about ourselves, those of us that are here in a moment. And I just also wanted to highlight our primary administrative assistant, Damian Rodriguez, who does amazing support for the department, both faculty and students. And he'll be the welcoming face you see when you first walk into the main office. And then Jamie Engel, who's been a biology academic advisor in the past, is going to continue to work with advising and support our students as well as other students in the college in the future. While I've got the mic, I'll just talk briefly about my own teaching. So I primarily work with the general biology sequence. I do labs for 191, 192, and then also do lectures as well as the labs for 193. And I, in addition to that, have done some non-majors courses, including the Intro to Biology with Lab course, and then a course I developed on the science and art of vision and how our mechanisms of vision play into how we perceive the visual arts. That's another pet area of mine because I was an artist growing up. And then I also have an exercise physiology course that I'm really excited to be teaching in the winter. That's for both undergrad and grad students. And I got into the interest of exercise physiology primarily because I'm a runner. So if that's something, if you're into biology and athletics, it's definitely a good course for you. You can even earn extra credit by running a 5K in the middle of it. And then I've also just got a picture of my own personal science experiment here. So I have twins, 7-year-olds now at home, and they've been an amazing learning experience in terms of, is it nature, is it nurture, or is it neither? And that's kind of the conclusion I've come to. But, and you're welcome to contact me. My email's here if you have any interest in learning more about these courses or potentially getting involved with materials prep for general biology in the future. And we have a couple other lab coordinators and teaching faculty with us as well. So I'll turn it over now to Rima Barkowskis.
Hi all. So let's see, here we go. Okay, so welcome. My name is Rima Barkowskis. It was kind of a crazy ride for me to get to the meeting here. I was driving to pick up a sick nephew for my sister-in-law. So, but I'm here, I'm making it. I can introduce myself. Hi, everybody. I recognize a few of my Bio 193 students, which is one of the courses I teach. So I do the off-sequence 193. In the past, I've also taught a junior year experiential learning class called Bio 302. And I also did anatomy one time. I tend to do a lot of cell bio and genetics lab coordination. So that's usually my big workload over winter and spring quarter. So if you end up in one of those classes, you are highly likely to run across my face again. Just in general, where did I do or what kind of research did I do? As a graduate student, I looked at aquatic physiology, looking at stress tolerance in some isopod and amphipod freshwater species of crustaceans. I've recently moved offices, so I hope this is the right office, but I do know it's within room 118, the main bio office. But soon after moving in, we got kicked out because of COVID. So that's why I can't ever remember it right now. I'm not used to going to that room. And I know we have lots of different concentrations within the bio majors. And so the one I'm most identified with typically is physiology, but if you're ever curious about talking about concentrations or what classes you want to take, I do advising over the summer. I took this picture a while back, pre-pandemic and pre-broken ankle, but I do enjoy biking and triathlons, and I have a couple fur babies that were definitely excited when I was home with them over isolations and things like that. So I look forward to meeting everybody, and I want to wish everyone a wonderful fall quarter.
All right, thank you very much. Claire Behrens.
Okay, I'm going to share my screen. Okay, can everybody see that? All right, so hello everyone. My name is Claire Barron. So I'm one of the lab coordinators and lab instructors here at DePaul. So I've actually been a lab coordinator at DePaul since 2012. And before that, I received my master's degree here at DePaul as well. So I'm a lab instructor for Bio 191 in the fall, and then I also coordinate the off-sequence General Bio 191 and 192 labs during winter and spring quarter. I was also privileged to be part of 193 labs for the first time last spring, which was really exciting. It was a lot of fun. So my research focus for my master's was on species diversity and ecosystem restoration. So I look specifically at how microarthropod diversity is influenced by above ground plant diversity, and then also, of course, soil health. So I wanted to share a current photo of myself with my family. So you can see I have a 3-year-old daughter there, Ariana. And then my new addition, Tyler, he's only 14 weeks old. He's really cute, but not so cute at 3 a.m. lately when I haven't been getting much sleep lately. So those two keep me quite busy. And then I had to show my two troublemakers, Remy and Logan. Just like Rima mentioned, her dog, my cat was ecstatic that I was home for 18 months with the pandemic. He was on many Zoom bombing DePaul biolabs. So, but so if we've not already been in lab together, I look forward to getting the chance to work with some of you in the general biology sequence soon. All right, thanks, Carolyn.
Thank you, Claire. All right, Dr. Megan Scrimenti. I think we may have missed her. She was with us earlier, but it appears that she is no longer. So she may had to have taken off to pick up her kids as well. But we do have Tim Sosa.
Hello, if there's anybody left, I'm on a computer without a camera, so I don't have a PowerPoint for you.
45 of us.
Huh? No, but no students left. No, I'm sure that's not true. I'm sure there are many of you left. You're most likely to see me in the intro biosequence, especially 192. I do also help with 193 from time to time. And I've also been involved in 215, which is ecology, and 201, which is human anatomy. And there's a couple of non-major classes I teach, and some other ones that I might be developing in the future. But those are the places you're most likely to find me. And so I did my undergraduate degree at the University of Minnesota in ecology and evolution and behavior. And I did a PhD at the University of Chicago in evolutionary biology. So that's, I'm mostly focused on the evolution of fishes, but I've also studied a lot on mammals and insects and a little bit on plants. So I'm just really into organisms. And so, you know, if you ever have questions about that kind of thing, I'm happy to help with that. I don't do a lot of research these days. I do a little in the summer, but my entire job is teaching. Like the rest of us here at the end of the meet and greet session. And so, you know, my door's open if you need help with classes, even after you've gotten out of my class. All right, I'm here as a resource. Looking forward to working with all of you this year.
So another really important part of our teaching at DePaul are the master students that support so many of our classes. So there's several of them here today, and I wanted to give them a chance to introduce themselves as well. So Kaylee.
Hello, can everybody hear me? All right, here, I wrote an introduction. Hello everyone, my name is Kaylee Arnold. I am a TA for the evolution class. And I'm a part of Dr. Aguirre's lab. My general interests include studying evolution and adaptation in response to the environmental changes coming about in the Anthropocene. Specifically, I'm interested in studying the effects of freshwater acidification on fish species. I did my undergraduate at Loyola, and now I'm here for 2 years, so yeah.
Okay, Cesar.
Hello. I hope everyone can hear me well. My name is Cesar Fuentes. I'm part of the Aguirre Lab too. I'm a second-year grad student now. I'm actually studying tropical fishes. I'm studying ecology of 2 species of cichlids in Guatemala, where I'm originally from. In Central America, and I'm very glad to have you all, and probably going to be seeing you throughout different labs and around the McGowan South and North.
Okay, James.
Hi, everyone. My name is James Hock. I'm a first-year master's student, and I TA a couple of different sections of Bio 191 this quarter. I work in Dr. Vistrianski's lab. I'm really into ichthyology, study of fish, and also looking at their cellular processes like salt regulation, osmoregulation, stuff like that. But I would like to introduce microplastics into the equation and see how that is potentially adversely affecting our fish populations. So that's what I'm interested in. And I graduated from a really small college in Alliance, Ohio, only about 2,000 people, University of Mount Union, but 13-time D3 national champions. So we're all very proud of that. And I had to give them a shout out.
Nothing wrong with D3. Go Jumbos. I went to Tufts. David?
Hi. I'm a second-year graduate student in Dr. Lamontagne's lab. My work is focusing on tree cavities and bird populations in the city of Chicago, but I'm also helping out with some of Dr. Lamontagne's tree reproduction work during the field season. This quarter, I'm TAing ecology, which is also my main focus in general. So most of my work is about ecosystems and kind of how they react to different inputs and processes.
Thank you. John?
Hi, I'm John Juranic. I'm a second-year grad student in Dr. Shimada's lab. Right now, I'm working on a bunch of this fossiliferous material. It just kind of looks like sand or dirt, but there's a bunch of shark teeth, fossil shark teeth and fish teeth in here. So I am sorting that out. This came from Nebraska, so sorting it into vials. And these are sharks from 100 million years ago that we are looking at their populations.
Thank you. Olivia?
Hi, my name is Olivia. I'm a first-year student in the Aguilar lab. I hope to work on the effects of climate change and just human impact in general on a species of fish called the blunt-nosed knifefish in Ecuador. And then hopefully I'll just be studying just general morphology as a result of climate change effects, and then just potentially maybe behavior after the fact. My previous research was in my undergrad at Auburn University. First, I got an undergraduate degree in organismal biology, ecology, evolution, and behavior, and then I got a second degree in animal sciences with a minor in natural resource ecology. And I worked at an evolutionary ecology lab that was ran by Dr. Warner. And so I worked with Anolis sagrii, which is a brown anole native to Cuba. And so what I looked at was just different incubatory conditions and how it affected general morphology, survivability of the embryos, and then overall, just how it affected their ability to just survive after the fact. And then I also looked at heritability. So I looked at the dorsal patterns that came from both paternal and maternal lineages, just to see how they would be inherited to the offspring. And so I'm really excited for this semester. I'll be TAing Bio 193. And so I've probably had I'd say most of you in lab already, but I look forward to getting to know all of you and working with you this semester.
All right, thank you. Teddy?
Hi everyone, I'm Teddy. I'm a second year master's student. I work in Dr. Lamontagne's lab on the spruce project. So I look at the effects of climate change on conifer reproduction. So I look at cone characteristics, so like cone length, cone width, seed viability, and essentially just see what climate change is doing to cones, because cones are super important for forest regeneration and composition. I'm looking at 2 species, so black spruce and eastern tamarack. They're very important in the boreal zone, especially in the peatlands. And currently I TA Bio 193. I've done ecology before, as well as Bio 192. And yeah, I think I've had some of you in lab before, and I'm excited to get to know the rest of you this quarter.
Are there any other master students here that I missed? Tried to search through the participants. I got all my RSVPs. Okay. All right, so I'm just gonna very quickly, I know it's getting late and you guys all have a Friday to get to, but I'm gonna very quickly go through just a couple more slides about other things going on in the department. So you've heard mention of a couple of these things already. I know Dr. Lamontagne mentioned that she's the faculty advisor for DEEP, or the Evolution in Ecology and Physiology student group. We also have a number of other active student groups on campus. We had a predental club, an animology club, global brigades. I'm actually the faculty advisor for, and they organize medical brigades, dental brigades, public health brigades, which have kind of, again, been put on hold temporarily, but will no doubt pick up as we start to travel again. And there's a pre-med club. There's also some student affinity groups that particularly focus on certain student populations and making sure that they have a home in the department where they feel welcome and are able to fully express their identities and share their experiences with other students. So we've got LGBTQ club, we've got a Women in STEM club, we've got a Diversity in STEM club, and then SACNIS is a very active group at DePaul as well, and that's not just within our department, but also throughout the college. I'll also post these slides on the seminar page along with a link to this video after the fact. And you can also find most of these organizations on DHub, and you can find contact information and meeting information there as well. And then just wanted to give you a quick overview of some of the support systems that are present at DePaul that you may not have heard of yet. So if you guys, especially the freshmen that are just coming in, and you know, it's a big adjustment coming to college. College is definitely not high school, either academically, personally, emotionally. So there's a lot of support systems that DePaul has in place to make sure that you succeed and thrive in your first year here, including the Dean of Students office, which is really important when you have to arrange absences from your courses or get in touch with some resources if there's something you're in need of. There's university counseling services if there's anything that you're struggling with in this really challenging time. The Office of Multicultural Student Success particularly supports groups that have been underrepresented in STEM in the past. And then the Vincentian Assistance Fund is available if students are dealing with periods of financial hardship. And again, you can find more information on a lot of these different support systems through the main DePaul website and the College of Science and Health website. Also wanted to let you know about some research opportunities that you can get involved in very early on. So if you end up doing research in any of the labs at DePaul or any partner institutions, there's this great research showcase every year called CARS, Chicago Area Undergraduate Research Symposium, that involves several other universities in the area where students present posters or talks on their research. DePaul also has our own in-house undergraduate research showcase every year for all the departments in CSH. And then there's also some financial support for some of this work. So there's undergraduate research or UREP fellowships that you can apply for with a faculty sponsor. And then there's also this Alice Amper Lewis Stokes Alliance for Minority Participation, specifically again to support research for students that are coming from a background that's been underrepresented in STEM in the past. And then here's just a little bit of where you can find the DePaul Bio Department online. So we've got a Facebook page, we've got a Twitter account, an Instagram account, there's a blog, and Damian Rodriguez, the main administrative assistant, is the person that you should contact if you want to get any media posted in any of these places. And then last but not least, just a quick plug for some more upcoming events that the seminar committee is putting on. So we'll have some in-person seminars this year, as well as some additional virtual events that we hope you can attend. We've got a new seminar component coming in this year called Journeys, where faculty and alumni are going to talk a little bit more about how they got to where they are today. So not just the science they're doing right now, but all the decisions they made and opportunities they had and challenges they faced as they were coming along in their careers. And then we'll have a special celebration for Darwin Day, and at the end of the year we also highlight our graduating seniors with a senior symposium that includes both talks and posters. Okay, So thank you all for coming today and for spending so much time with us. I hope you got to know our department a little bit better. And like I said, I'll definitely post these slides. I'll also post this video on the DePaul Biological Sciences seminar page, and we also have a seminar YouTube channel. So if you would like to watch any of this again or share it with any of your friends, I'll definitely make that available by early next week.